In this video, we will discuss about C3 cycle. C3 cycle occurs in plants which do photosynthesis. What is photosynthesis? It is synthesis of carbohydrates or sugars in presence of light. This is called as photosynthesis. So during photosynthesis, there is a mechanism called as C3 cycle. Now, why this is called as C3 cycle? Because first stable product which is produced during this cycle is 3 carbon in length called as 3-phosphoglyceric acid. So we can call this is COH, CHOH, CH2OP, 1, 2 and 3, 3 carbon in length. So this is called as C3 cycle. Now this was discovered by scientists Kelvin and Benson. So this is called as Kelvin-Benson cycle. And plants in which this cycle is operative, those plants they are called as C3 plant. Another name for this cycle is reductive pentose phosphate pathway. Then this is also called as PCR cycle, photosynthetic carbon reduction cycle, because here carbon is being reduced and carbohydrates are being produced. So this is about C3 cycle. Now this is called as dark reaction because this reaction is not occurring in dark, but this does not require light for this reaction. So this is called as light independent reaction. And this is happening in the stroma of the chloroplast. So this is chloroplast, which is double membrane enveloped. And this is the matrix or stroma of chloroplast. So here, this cycle is operative. So this is called as matrix or stroma of the chloroplast. So this is happening here in the matrix or stroma. Now this light reaction is taking place in the granum part. That we'll discuss in the next video. Now this C3 cycle or dark reaction or light independent reaction, how does this occur? This is actually completed in 3 steps. First step is carboxylation, as the name indicating, which is leading to fixation of carbon dioxide. Carbon dioxide will enter into cycle and that will be fixed. Then glycolytic reversal. In glycolysis, what happens? Starch get converted to pyruvic acid. And when this is reversed, right, this is called as glycolytic reversal. So formation of sugars, glucose, fructose, sucrose occurs during this cycle. Hexoses mean they are 6 carbon in length. Sugars, they are 6 carbon in length. So they are called as hexoses, like glucose, fructose. They are 6 carbon in length. Then regeneration of ribulose 5-phosphate. And this is being regenerated at the end of the cycle. So 3 steps are there. Number 1, carboxylation. Number 2, glycolytic reversal. Number 3, regeneration of ribulose 5-phosphate. Enzymes which plays an important role in fixation of carbon dioxide in the C3 cycle. First enzyme is Rubisco. Now what does this enzyme say? Its full form is ribulose 1,5-biphosphate carboxylase oxygenase. If carbon dioxide concentration is high, then this part of the enzyme, ribulose 1,5-biphosphate carboxylase, will become activated. This will help in fixation of the carbon dioxide. But if carbon dioxide concentration is low, then ribulose 1,5-biphosphate oxygenase part will be activated, and that will help in photorespiration. That we have discussed in another video. Link will be shared in the description box. So this part of the enzyme will be activated, that is ribulose 1,5-biphosphate carboxylase, when carbon dioxide is high, temperature is low, and proper moisture is available, optimum amount of turgor pressure is there, moisture is there, and plant is healthy, it is not suffering from any diseases or water loss, then photosynthesis will happen, and that will happen with the help of, or fixation of carbon dioxide will happen with the help of this enzyme, ribulose 1,5-biphosphate carboxylase. Now we'll discuss cycle in detail. Now first acceptor of carbon dioxide, which accepts the carbon dioxide, is the ribulose 1,5-biphosphate. Now this ribulose 1,5-biphosphate, as the name indicating, this is 5 carbon in length. 1, 2, 3, 4 and 5. At first position there is phosphate and second position there is phosphate. Now this is being converted from ribulose 5-phosphate. Now this is ribulose 5-phosphate, where only at the 5th position phosphate is there. Now this will get converted into ribulose 1,5-diphosphate with the help of ATP and ADP. Because we are taking here 6 molecules, so 6 molecule of ATPs they are required. Now ATP will give phosphorus to this ribulose 5-phosphate and will get converted into ribulose 1,5-biphosphate. Now this ribulose 1,5-biphosphate, which is an acceptor of carbon dioxide, now this carbon dioxide will be fixed with the help of this enzyme. Rubisco, we have discussed already, ribulose 1,5-biphosphate carboxylase, and magnesium ions are also required for its activity, and this step is called as carboxylation because carbon dioxide is being fixed. So we can see here carbon dioxide is added here. So this is ribulose 1,5-biphosphate and here carbon dioxide is added. Now when this carbon dioxide has been added, this becomes 2-carboxy, 3-keto, 1,5-biphosphorybital. Now this product in presence of water, this will break up into, from here, this will break up into 3 carbon compounds. Like this is 3 carbon, this is also 3 carbon because it was 6 molecules. So 6 molecule total will be 12 molecules. So we'll get 12 molecules of 3-phosphoglyceric acid by its breakdown, right? COH, CHOH and CH2OP, right? Now this 3-phosphoglyceric acid, now this will get, because this is the first stable product which is being produced and this is 3 in length, so this is called as C3 cycle. So sometime one number question may be there why it is called as C3 cycle. I hope this is clear to you. Now this 3-phosphoglyceric acid, again this will be converted to 1,3-biphosphoglyceric acid. Now here at 3 position phosphorus is there, at 1 position this will be added from the ATP. ATP is what? Adenosine triphosphate. So there are 3 phosphates, one will be added here over here and this will get converted to 1,3-biphosphoglyceric acid and this itself will be converted to ADP. So because 12 molecules are there, so 12 ATPs they are required. 6 we have already discussed. These are 12 molecules, so total will be 18 molecules, right? Now this 1,3-biphosphoglyceric acid, now this will be reduced in presence of glyceraldehyde 3-phosphate dehydrogenase, and hydrogens will be donated by NADPH2. This will donate the hydrogen and this 1,3-biphosphoglyceric acid, this will be converted to glyceraldehyde 3-phosphate. Here this group will be converted into CHO and this phosphorus will be liberated and this will be accepted by water and this will form phosphoric acid H3PO4 and NADPH2. After donating hydrogen, this will get converted to NADP. So we'll get 12 molecules of glyceraldehyde 3-phosphate which has an aldehyde group. Now here we can see, now this glyceraldehyde 3-phosphate, out of 12 molecules, 5 will be isomerized. So this is called as isomerase enzyme and this will get converted to dihydroxyacetone 3-phosphate. Out of 12, 5 will move for the formation of dihydroxyacetone 3-phosphate. So 7 will be left now here. Now this 5 molecule of dihydroxyacetone 3-phosphate, they have a keto group. Here this was aldehyde group. Now out of the 5 molecules, 3 molecule of dihydroxyacetone 3-phosphate and 7 was left. Out of 7, 3 will move here. So total now 4 will be left here, right? So 3 molecules of glyceraldehyde 3-phosphate, 3 molecule of dihydroxyacetone 3-phosphate, they will condense to form 3 molecules of fructose 1,6-diphosphate, like 3 molecules like this we can see here, 3 and 3, right? This will remain 3. So this will become 6 carbon in length at first position phosphate and second position phosphate. So this is fructose 1,6-diphosphate. In presence of phosphatase, now this will lose this phosphorus, will get converted to fructose 6-phosphate. Right? Only at the 6th position phosphate is there. So this is fructose 6-phosphate. Now out of the 3, 1 will lose its phosphorus to the first position. This is called as isomerization. Isomerase will help conversion of fructose 6-phosphate to fructose 1-phosphate. So this is fructose 1-phosphate. Now this fructose 1-phosphate, this one molecule will enter into formation of sugar. Now this will be converted to glucose 1-phosphate. Now glucose 1-phosphate will lose its phosphorus and get converted to glucose. Now this glucose acts as a precursor for the formation of sucrose, starch and other types of sugars. And how this is happening, we'll discuss just now. You can see here, glucose 1-phosphate reacts with ATP This enzyme required is ADP glucose phosphorylase, and this will help in conversion of ADP glucose. Glucose 1 phosphate can also react with UTP, that is uridine triphosphate, and enzyme required is uridine diphosphate glucose phosphorylase. This will get converted to UDP glucose. Now ADP glucose will provide glucose residues for the formation of the starch. Starch is a polymerized product which is being produced by polymerization of many glucose molecules. Now starch can be produced. But sometimes UTP glucose and fructose 6 phosphate, they may condense with each other. They will combine to form a molecule called as sucrose. Now sucrose is the first stable sugar produced during photosynthesis, and because chloroplast wall is impermeable to sucrose, so this will be loaded in the phloem tissue from where it can be transported to other parts of the plant. So this is how sugars are being produced during photosynthesis, as the name is indicating. Synthesis during light, and this synthesis is of carbohydrates or sugars. So this is the product of the photosynthesis which is being utilized for the plants for various activities, as well as this is also being stored in the form of starch as a reserve food material. Now we were here 3 molecule of fructose 1,6 diphosphate, they get converted to 3 molecule of fructose 6 phosphate. 1 molecule goes for the formation of sugars. Now here 2 will be left, right? Now 2 will move here. 2 molecule of fructose 6 phosphate. Now here we have seen that out of 12, 5 moved here, 7 left. Out of 7, 3 moved here, 4 left. Now out of 4, 2 molecule will move here, right? And 2 will be left. So 2 molecule of glyceraldehyde 3 phosphate and 2 molecules of fructose 6 phosphate, they will combine, right? So this will become 9 carbon in length. This is 6 carbon. Now 3 carbon will be added, and this will be 9 carbon in length. So now this will break up here, right? 1, 2, 3, 4, 5, 6, 7, 8, 9, right? So this will break up 4 here, 5 here. So this will break up into 4 and 5 carbon compound. So this is 4 carbon is erythrose 4 phosphate and 5 carbon is xylulose 5 phosphate, because 2 molecules were condensing. 2 molecules, so 2 molecules of 5 and 2 molecules of 4 will be produced. So 2 molecule of xylulose 5 phosphate, like we have shown with the orange color, these 2 molecules are coming from here. So 2 molecules of xylulose 5 phosphate and 2 molecule of erythrose 4 phosphate. One is having keto group and second is having aldehyde group. Enzyme required is trans ketolase, thiamine pyrophosphate and magnesium ion. So this is the formula of xylulose 5 phosphate, CH2OH. This is keto group, and here this is aldehyde group. Now this 2 molecule of erythrose 4 phosphate, which are 4 carbon in length, having aldehyde group. Now this will combine here with the molecules of dihydroxyacetone 3 phosphate. So 2 molecules of this and 2 molecules of this, right? Now this is 4 carbon in, right? So this is 1, 2, 3 and 4, and this will combine with 3 carbon, and this will become 7 carbon in length. Now because phosphate will be present at 2 positions, so this is called as pseudohaptulose 1,7 diphosphate. So this will be produced after condensation, and enzyme required for condensation is aldolase. Now pseudohaptulose 1,7 biphosphate, this will lose its phosphorus and will get converted to pseudohaptulose 7 phosphate. At the 7th position only phosphate is there, right? So the enzyme required is phosphatase, which will remove the phosphorus, and this will get converted to pseudohaptulose 7 phosphate. So 2 molecule of pseudohaptulose 7 phosphate will be produced. Now these 2 molecules of pseudohaptulose 7 phosphate, like we discussed here, out of 12, 5 moved here, 3 moved here and 2 moved here. 5 plus 3 plus 2. Now 10 molecule. Now 2 will be left. Now 2 molecule will move here, right? And they will condense with pseudohaptulose 7 phosphate, 2 molecule of this and 2 molecule of this, they will condense, and this will become 7 carbon in length. 3 carbon will combine, this will become 10 carbon in length. 1, 2, 3, 4, 5, 6, 7, 8. I'll again make 10 carbon. 3 to 5, 6, 7, 8, 9, 10, right? Now this will break from here, 5 carbon. So 5 carbon will be produced. So we can see this will break up into 2 molecules of ribose 5 phosphate. This is 5 carbon in length, and 2 molecules of, again this will break up to form 2 molecule of xylulose 5 phosphate. 2 molecule from here and 2 molecule from here. Total this has been shown with yellow color, which are coming from here. So total will be 4 molecule of xylulose 5 phosphate. So 2 molecule of ribose 5-phosphate and 2 molecule of xylulose 5-phosphate, they are produced. Now this 4 molecule of xylulose 5-phosphate, they will undergo epimerization, and this will get converted to ribulose 5-phosphate, right? So from here 4 molecule of ribulose 5-phosphate are being epimerized, right? And here 2 molecules of ribose 5-phosphate, they will get isomerized. So 4 from here and 2 from there, total molecule will be 6 in number. So 6 molecule of ribulose 5-phosphate will be produced. Now this has been regenerated. So this is called as regeneration of ribulose 5-phosphate. So ultimately this will get phosphorus from the ATP, and this will again get converted to ribulose 1,5 biphosphate. So in this way cycle is completed and there is formation of the sugars. So as we discussed, there are 3 steps. Number 1 is carboxylation. Another is glycolytic reversal. So we can see this is glycolytic reversal leading to formation of sugars. And another is regeneration of the ribulose 1,5 biphosphate. And this is being done in a cycle. So this is called as C3 cycle, where first stable product is C3 in length. So in this way there is formation of sugars. And we have also seen how these sugars, they are being produced during photosynthesis. So synthesis of carbohydrate is taking place. So overall reaction will be 6 carbon dioxide, 18 ATP, 12 NADPH2, formation of sugar, 18 ADP, 18 inorganic phosphate, and 12 NADP. So 18 ATP and 12 NADPH2 are being utilized in the cycle for the formation of the sugar or carbohydrates. So this is all about C3 cycle. Now we can see here how this is 18 ATP. See here there are 6 ATP molecule required. Here there are 12. So 12 plus 6 is equal to 18. And here 12 NADPH2 they are required. So 12 NADPH2 and 18 ATP are required for the formation of the sugar. So this is first stable product, which is C3 in length. So this is called as C3 cycle. And plants in which this cycle is operative, they are called as C3 plants. And this reaction is taking place in the matrix of the chloroplast. So this is all about C3 cycle. If you have any doubts or questions, you can ask me in the comment box. Thank you for watching my video. If you like my video, please like, share and subscribe.